Alright guys, so by now you should have filled up your handy dandy study guide. We're going to go over most of the answers to these so that you are prepared for your DNA test and can be really successful. This time I'm not giving you a key to copy down, so you're actually going to watch this video and pay attention, but we've already gone over all of this already, so you should know it and be amazing. First off, you have to know what DNA stands for, which is deoxyribonucleic acid. We know that our DNA molecule is made up of several different parts. The sugar phosphate backbone is this part on the outside right here, and then on the inside we have our nitrogen bases, and these match up with each other and we call them our base pairs. This whole little section right here, if we zoom in, we have what we call a nucleotide. The base is going to be right here, either A, T, G, or C, adenine, thymine, guanine, or cytosine. We have a sugar connected to it, and then a phosphate right here. And again, all three of these together is our nucleotide. So make sure you don't forget your base pairing rules, which are going to be pairing up your nucleotides G to its pair C and A to its base pair T. And so there's a couple mnemonics you can use to remember these. Some of the ones you guys thought of were all teachers can go. Whatever works in your mind, do not forget your base pairing rules. Okay, so next you're going to have to be able to identify where the DNA is located in the cell, which is the nucleus. So that's just going to be right in there. Alright, so next we're talking about DNA and chromosome structure. Remember, your chromosomes are made up of your DNA. If you took any little cell in your body, you'd look inside the nucleus. Inside that nucleus would be a bunch of jumbled up DNA called chromatin, but when the cell is ready to divide, it condenses into those chromosomes, which look like these funky butterflies that we've seen. And remember, the DNA in chromosome form is wrapped up around lots of proteins. It's very, very condensed, so that's what we see when we do our karyotype, which we'll talk about later. Alright, DNA replication. Remember, this is the part when our cell is going to copy the DNA to get an identical set of it before cell replication or cell division. So, DNA replication is the DNA is going to have to copy itself to be completely prepared for mitosis. So, the DNA has to be completely replicated in the S stage of interphase, which is DNA synthesis, in order to start mitosis. So, the steps of DNA replication in its basic form, we've got another video for this, so if you don't really remember that, you might want to go through and review it, watch the other video, but this is just the basic outline of what's going to happen in DNA replication. First, an enzyme, remember it's an enzyme because it ends in the letters ASE, called helicase, will unzip the DNA molecule. It will cut right up the center of it, and then we have an open side and another open side. Then we have free floating nucleotides that are going to match with their base pairs, and so we have one side being built new and the other side being built new as well. And then polymerase, another enzyme, will come and join together the new backbones until we have what's called semi-conservative replication, where we have two exactly identical new molecules of DNA, each with half old and half new sides or strands. Next up, we have to talk about part of the cell where DNA replication occurs. Of course, just like we said, DNA happens in, or occurs in the nucleus, is in the nucleus, DNA is there, so replication occurs there as well. So you're going to have to be able, if I give you the original strand of DNA, you're going to have to be able to match up the complementary strand with their base pairs. So it's going to match up nicely like that. The A to T, T to A, G to C, C to G, C to G, and T to A. Alright, protein synthesis, my favorite part of DNA, because we start to involve RNA, which is another nucleic acid. RNA is ribonucleic acid. Now, the sugar is a little bit different in this molecule. It's a ribosugar instead of a deoxyribosugar, so that's why it has a different name, but the basic structure is the same. There's a few key differences, though, that we want to talk about. DNA is double-stranded. You can remember that with the DNA starts with a D, double starts with a D2. RNA is single-stranded. It's all alone by itself. But RNA can travel in the nucleus and out of the nucleus. It's special like that. DNA, always in the nucleus. Remember, DNA uses the base thymine. RNA does not. Anytime we would see a T in DNA, we now replace it with a U for uracil in RNA. And finally, there's three key types of RNA: mRNA, tRNA and rRNA, messenger, transfer, and ribosomal RNA, which we'll talk about. So these three big guys are messenger, because this one is the one that's going to be copying the original DNA strand, sending that message out of the nucleus. Ribosomal makes up the ribosome, easy enough. And transfer RNA is going to transfer over that amino acid so we can build our protein. Now our main steps of protein synthesis, I hope you have not forgotten, because this is the central dogma of molecular biology. This is how everything in our body is going to happen. We have DNA turning into RNA, turning into a protein made of amino acids. Don't forget it. So the process of DNA to RNA is called transcription. The process of, transcription. The process of RNA turning into a protein or being changed biochemically into a protein is called translation. And the way I like to remember this, I think of in the nucleus, somebody transcribing, writing down really, really fast, just a general copy, one nucleic acid to another. But when we're changing biochemical languages, we're going from a nucleic acid to an amino acid. It's like going from French to Spanish, so we have to translate it. We're turning it into a different language. So when we do the first step, transcription, remember that occurs in the nucleus, we're going from DNA to RNA. And when that happens, remember we replace anytime we would normally put a T with a U, we just pair up the regular base pairs, and there we go. Remember, there's a full video of protein synthesis practice which we have, which you should watch if you're still confused, but you guys all did a great job practicing it in class already, so I think you're experts. We also need to talk about translation, which happens outside of the nucleus. Remember that mRNA is going to travel after it's copied the DNA out of the nucleus. It's going to meet the rRNA, it's part of the ribosome, and then this tRNA molecule will match up with every codon, having its anticodon here to connect, and then bringing over the amino acid that will link up and eventually give us a beautiful protein. So lots of amino acids chained together will give us our protein. Amino acids are going to be one of the 20 essential ones. It depends on what the codon is to figure out what the amino acid is. Remember, we use our codon chart to figure that out. And a bunch of amino acids all folded up together nicely are called a protein. At the start of our protein, we will have a special start codon that will generally give us this particular amino acid here. And at the end, we have a stop codon, which tells our cell, our process of protein synthesis to stop happening, so we don't keep coding proteins forever and ever. A codon is just a group of 3 bases together, like UAU, and then we can match it up with a specific amino acid on our codon chart. An anticodon is the opposite side, the complementary side, the 3 opposite bases that the tRNA will have, so it knows what to match. Now, we've talked about this a lot, but the fact that all of our bodies and all of our cells are going to do this is really important. Every single process we have in our body is pretty much controlled by proteins. Everything from the color of your eyes down to how your cells transport oxygen is going to have to do with proteins, so we need to do this every single day. And non-functioning protein synthesis is going to give us diseases and lots of problems. Now let's talk about mutations.
So this is going to be under topic 4. You're going to have to identify a gene mutation as opposed to a chromosomal mutation. So gene mutation is just going to be mix up just in a single gene, whereas in a chromosomal mutation, you're going to have you could have an entire extra copy of a chromosome, or you could have one too few, whereas here in this example, you have 3 copies of chromosome 21, which is called trisomy 21, also known as Down syndrome. So we're going to talk about some different types of gene mutations: substitution, insertion, and deletion.
Substitution is when we substitute one base for another, so potentially here there might have been a G or a T or an A, but all of a sudden, oops, there's a C. And as we saw when we were doing protein synthesis, this could cause us to have either a totally different protein, or it could just swap out an amino acid. So it could be a big problem, it could be a little problem. We really have to see the protein in order to figure out what exactly went wrong. Insertion, though, and deletion, these are bigger issues. These are called frame shift mutations, meaning that they're going to shift over everything that follows the mutation or push it back. And that's going to give us a big issue because every single codon that follows will be screwed up, and then every single little amino acid we messed up will have a protein that doesn't work. We call it a non-functional protein. So in an insertion, we're adding, it sounds like what it is, we're adding an extra base somewhere where it shouldn't be. In a deletion, we're getting rid of a base, and then we have everything shift over by one, screwing up our entire protein synthesis process. So we can have positive mutations as well, and these can provide us with different traits as we've seen in evolution, and sometimes those traits are beneficial. Those species are able to outcompete, to survive, and to reproduce more than other individuals, and thus we have a wonderful thing of natural selection and survival of the fittest happening. Sometimes mutations can be terribly non-beneficial, and these mutations will cause organisms to die early or potentially have traits that are non-beneficial and cause them to not survive and work well. So we'll talk more about our mutations and our problems when we get to our genetics unit.
Alright guys, hopefully that gave you a pretty comprehensive look into your review topics. Make sure you study hard. I think you can rock this test. If you need any extra reviews, go back and watch those old videos I mentioned. You can rock this test. See you next time! Alright.
Do you know the differences? You can skip that.
Oh yeah, I haven't done that since freshman year.
Can you describe protein synthesis? And then in the ribosome, the transfer RNA matches up with each of the codons.
You might just want to use your part
Yeah,
for this.
Okay.
Oh, I can talk about this.
Okay.
We did
Okay.
karyotypes and stuff.
Okay.